I'm Audrey Tremolay. I'm one of the pediatric doctors. So these doctors will know me as the Kawasaki, one of the Kawasaki doctors here. Thank you for joining us. And I have the distinct pleasure of introducing a close colleague and friend of mine, Dr. Jennifer Lay. Jennifer is a PharmD by training. She did her undergraduate degree at UCLA and then did her degree in pharmacology up at UCSF. She's also then done on here at UCSD to do a really great program, a master's in clinical research. So I highly recommend everyone interested in clinical trial research. We talked to Jennifer and know a little bit more about that. And really what I have found as a colleague with Jennifer that makes her so special is that between my part of clinical trials and my interest in drug dosing for children, she's the one that makes the magic happen with understanding the pharmacokinetics and the pharmacodynamics, proper dosing, side effects and adverse events. And her work has really moved forward the field of many antibiotics, including a lot of what I'm sure she'll speak about today with vancomycin. So thank you and help me welcome Jennifer.
Thank you. Thank you for that wonderful introduction. So I have no conflict of interest related to the presentation today. However, at the end of my presentation, I'm going to share some of the research that I was involved in, and that was funded by the NIH through the K23 award. So here's the structure of what I'm going to go through. I divided the session into three main segments. First is just going back to the basic. What is clinical pharmacology? And how do pharmacy in particular play a role in this? From the patient care side to the research as well as education. And I'm going to go into key terms that I hope you'll remember after the session. And this includes population-based pharmacokinetic modeling, Bayesian estimation, and Monte Carlo simulations. Then I'm going to proceed into bringing up the recent literature on vancomycin. Yes, it's a very old drug. It's like 50 years old, but we're still learning how to dose it properly, especially in pediatrics. So I'm going to review some of that literature that's come out in pretty much the last 5 years. And that's going to actually be reflected in the next national guideline that's due sometime this year. And lastly, I'm going to end with a case that I will share of a patient that we have and trying to illustrate the key things that we learned from research and bringing that into practice. And here are the learning outcomes. I think that's on the handout that you have. So let's start out with just basic what clinical pharmacology is. So the ultimate goal of clinical pharmacology is really to get effective, is to ensure effective as well as, especially for pediatric and safe pharmacotherapy. So how do we achieve this? And the way I like to explain this is really through the 3 Ps that affects effective and safe pharmacotherapy. And the first P is pharmacokinetics. This is essentially how our body handles the drugs. And this not just varies between pediatrics and adults, but it also varies between the various pediatric age group that we have, all the way from preemies, neonates, all the way up to adolescents. The second P is pharmacodynamics, which is essentially how the drug works on the body. Now, with pharmacodynamic, it does encompass safety as well, and this is very, very important. And as you know, for any drug to be approved, especially for use in pediatric, it has to have a safety evaluation first. And lastly is pharmaceutics, and this is oftentimes overlooked in the adult population, but it's very critical also. A component of effective and safe pharmacotherapy is pharmaceutic, and this encompasses the formulation, the inert ingredients that we think is inactive, but it may have some issues for pediatric use, especially in the neonate population, as well as the taste, and we all know that. And for formulation especially, I think we consider oral suspension, and I know all the pharmacists will hopefully agree with me on this, would be oral suspension is kind of like the holy grail for pediatric use of drugs. Now, there are major gaps in effective pharmacotherapy in pediatrics. Now, at the bottom of this, we all know and are aware that adults, pediatrics are not adults, and children just don't necessarily handle and respond to drugs as the way adults do. So extrapolating from adult pharmacology does not always work. Most of practitioners in pediatrics do know this cliche here, one size doesn't fit all, but when you don't talk to an adult practitioner, they will have no clue what we're talking about. And essentially, we cannot have one dose that's going to meet the care of the entire pediatric age group. We're going down from infants all the way to adolescents. So that's something to keep in mind, and hence having an oral suspension, and I know this is endorsed by the AAP and some professional pharmacy association, where we really need to advocate the need for oral suspension for any new drugs that are available so that we can customize the dose to meet the pediatric needs. Now, so far, I kind of defined what clinical pharmacology is. So who are the players in clinical pharmacology? And we have obviously physicians, nurses in this paradigm here. But what I wanted to particularly emphasize was the role of clinical pharmacists in clinical pharmacology. First off is as healthcare providers, and the term we use for this is therapeutic drug monitoring, the TDM. And I know at Rades, pharmacists are very involved. Gail, who's here, and I don't see Bill. Oh, there you are, okay. Who are very involved with that to ensure the safe and effective use of vancomycin and aminoglycosides here at Rades. The other aspect of the other role of pharmacists is through research, and we call this systematic clinical pharmacologist. And I'm going to spend a little bit more time to explain what this is, in particular explaining the tools that we have that we use to optimize drug dosing in pediatrics. And lastly is education as another role. Okay, for research, so how do we go about with conducting systematic clinical pharmacology research? The goal of this is to optimize dose, but also at the same time minimize sample size that we need to complete the research to get some meaningful data. So we generally start out with adult PK data because that's essentially the only thing that we have, especially for new drugs. And note that these are generally done in healthy volunteers with intensive sampling. Then we use any in vitro or animal models, particularly when we simulate the effect of drugs, the pharmacodynamic targets. So we integrate that in, and any other literature from companies or that's published. And then we use all of these facets of data to scale models to children, and we scale it by size and maturation, particularly for organ function there. And using this, we're able to conduct simulations to essentially estimate what the best dose could be, and then we use that information to conduct the clinical trials. So this provides us more of a systematic way to make the best guess of what the dose that we should use in clinical trials so we can conduct that more in a cost-effective manner. There are challenges to pediatric clinical studies, and recently there's been strategies, some of which are recognized by the FDA as ways to study pediatric drug dosing. And I provided 3 examples here of strategies to overcome the challenges. First is an opportunistic study design, and then with sparse sampling, we can use population-based PK modeling. Now, with dry blood spot technique, this is something that's quite innovative that's actually done in third world countries because there's no freezer or centrifugation that's available, where we get 20 times lower the amount of blood that can just go on these cards. You just put the drop of blood and you can assay for drugs on that card. For opportunistic study, what's valuable here is that you get very high enrollment rates. So essentially, primarily because you have very low risk to the patient. What this involves is really using the standard of care clinical indications of the drug, so the patient's already on it. Now it's just adding on samples that we need to better understand the drug. So because of this, with the high enrollment rate, we're usually able to get a wider range of age group in pediatrics, so allowing us to really understand the drug from more of a developmental aspect, all the way from neonates to adolescent. Coupled to opportunistic designs is population-based PK modeling, and this is a big term, and I'll highlight some of this that was used in recent studies in vancomycin that we published. But the FDA, as well as the European Medicines Agency, support the use of population-based PK modeling in pediatrics. This modeling technique allows us to actually quantify how the drug is handled in the body through estimation of volume of distribution and clearance. It allows us to also appreciate the variability between patients as well as within the patient. And this could be very useful when you have patients, particularly in sepsis, who have dynamic changes from day to day. Now, traditionally, PK studies were conducted like this, where we have intensive sampling at certain time points for each patient. What population-based PK is works more with sparse sampling. So you don't have to get 6, 7 blood draw in a patient. You can get 2. The only exchange with that is that you have to have a larger number of patients enrolled in the study, which you can get. But essentially using sparse sampling coupled to population-based PK, we can still get a good idea of how the drug is handled in the body. So that's population-based PK. The second big term I wanted to explain was Bayesian estimation, because this is going to be a big idea in the incorporated into the next vancomycin guideline. What that is incorporating data that we already know that exists, usually from population-based PK studies. We already know how the clearance is, how the volume is in that particular population, and how the variability is. Using that data, coupled to data that's specific in that patient, dosing history and observed concentrations. And the integration of these two data, we can perform Bayesian analysis and get what the best dose is for that patient and what the exposure would be for that patient. Okay? So this is Bayesian estimation. Non-Bayesian estimation generally only comes from this side, just dosing history and drug concentration in that patient without much regard for prior data. So when you combine Bayesian estimation with population PK, it brings more accurate quantification of your PK parameters. And hence, it allows us to really have more predictive statement, not just associate, predict how it would be in that population or in that patient. So it's quite a very powerful tool. Another tool that we use in clinical pharmacology research is Monte Carlo simulations. And what that essentially is just computer-generated patients that would reflect what it would be in real life. And in the study that we did a simulation of over 10,000 subjects, which we would never get in a clinical trial for pediatric. But what this does is that it uses what we already know and then allows us to predict what the best dose would be based on the target that we want. So it really integrates, especially for infectious diseases, the drug, the patient, and the bug, kind of trying to describe the interplay with it to give us what the best dose would be that we can use for further study. And one of the key things that comes out of Monte Carlo simulation is probability of target attainment. It's essentially is the percentage of virtual subjects that would achieve the target that we're looking for, mostly coming from the efficacy standpoint rather than safety. But we can incorporate safety if we wanted to. But most of the focus now is on efficacy of drug. I do want to highlight that research, especially for pediatric clinical pharmacology, is very abundant locally. In collaboration with Rady Children's and UCSD, we have a long-standing history of success in clinical pharmacology research in pediatrics. And this is through the NIH-funded Research in Pediatric Developmental Pharmacology, RPDP is how I'm going to name it going forward, that we have 3 investigators on this, Dr. Edmund Caparelli, Victor Nizet, and our own Dr. Tremblay here, who are involved with this, and we are one of 4 sites nationwide. So it's quite prestigious, difficult to get this award, but definitely prestigious once it's here. And the goal of RPDP is to advance the knowledge of basic and clinical pharmacology, PK, and clinical therapeutics of antimicrobials and anti-inflammatory agents. So the RPDP, we do have an active RPDP award for that now, but it has a long-standing history lasting over 2 decades. It started in 1994 with Dr. Caparelli being involved with this over 2 decades, 24 years, 1994, yeah, 24 years. Starting with, it used to be called the Pediatric Pharmacology Research Unit, PPRU, and it's slowly evolved now to RPDP. In addition, since 2015, because of the strength of RPDP, we're able to procure training grants. T32 specifically for students and T35 for postgraduates. So we're just getting stronger every day in this area, which is great to see. And here's just a tiny record of RPDP's record of success. And this is just from my side of the study, which I think I'm least productive of everyone else in the RPDP group. But this is just to kind of illustrate the research that we do within this unit. The first study that was there, we were able to show, at least from pharmacology side, to demonstrate either it's safe or effective. So with the first study, we were able to learn that antibiotics was the #1 drug class associated with adverse drug events in hospitalized children. And then from the next 2 studies, we learned that for an old drug, we are underdosing. We're not meeting the pharmacodynamic target that we should be, especially for invasive infections. And in fact, we published the largest study known in pediatrics related to vancomycin use. And this could have only happened with the collaboration of Dr. John Bradley at Rady Children's, as well as the pharmacies team, the stellar pharmacy team we have here through Bill Murray and Gail Romanowski. So thank you for that. And lastly, through collaboration with Dr. Tremblay here, we were able to, yes, it's an old drug, ampicillin's even older than, I think, vancomycin. But we were able to simplify the dosing regimen to make it more meaningful and easier to use for clinicians. And I believe this was submitted for product labeling change with the FDA. Okay, any questions so far on that? Now you know clinical pharmacology, Bayesian estimation, POP-PK, and Monte Carlo simulations. So what I'm going to do now is kind of mention those tools again that we use and apply it to our recent findings for vancomycin use specifically in pediatrics. Okay. The guideline that most hospitals still use right now on vancomycin was published in 2009. And at that time, all the literature and the recommendations that we have for that guideline came from adults. There was no pediatric data at all. In fact, if you read the guideline, it'll say pediatric data is lacking. Just consider this, not necessarily use. But because of limited pediatric data, most of the pediatric hospitals had to rely on what was done in adults and apply it in our population. And in this guideline, the AUC to MIC was the target attainment for pretty much as it was our efficacy surrogate. So AUC to MIC of 400 or more was what we aimed for. And this corresponded to troughs of 15 to 20 in adults. And so what we did with that was a lot of the different pediatric hospitals started chasing troughs, because that was the recommendation also in this guideline as to monitor. We're going to monitor trough and chase trough to 15 to 20. And so that's what we did. Recently, we've come to learn that, well, AUC to MIC of 400 more is more achievable in pediatrics, and I'll tell you what that means later with another slide. But actually, this target that we know for efficacy is actually closely related to trough concentrations of 9 to 10, not 15 to 20. And so you can see the issue here is that since we've been chasing troughs of 15 to 20 in pediatrics, we may have increased the dose unnecessarily in some of the patients who didn't meet these targets. So using Monte Carlo simulations with Bayesian estimation, we were able, in a virtual subject of 11,000 there. So what we were able to find that at a dose of 60 milligram per kilo per day, assuming an MIC of 1, an AUC or area in the curve of 400 really corresponded to troughs about 9. And this was a study that was done in pediatrics, so 3 months or older. And there was another study out from UCSF, he's at Stanford now, that confirmed the same thing for neonates. The trough was lower than, it was actually 7 to 11 for neonates. So here's a very important slide. This tells us 3 very important things here. Now, just to let you know, A here, A and B, this represents 2 different hospitals. A represents Rady Children's, and B, the other hospital that was involved in the study at Miller Children's Hospital in Long Beach. Now, A is yellow and B is blue, and it represents the PTA, the probability of target attainment, based on an AUC, assuming an MIC of 1, of 400 or more. So at a dose of 60 milligram per kilo per day, we're getting about 70 to 80% target achievement, okay? And what first we learn here is that whatever we achieve will vary by hospital. It will vary by hospital because of the MIC distribution. At Rady's, I believe our MIC distribution was, we had 15% at MIC of 1.5 or higher. Whereas the other hospital achieved a lower target attainment here because their MIC distribution was a little bit higher. It had 30% at MIC 1.5 or higher. So 1, each institution will vary because of the MIC distribution. 2, when you look at the achievement at AUC, it's much more achievable in pediatric compared to trough. We have about 70–80% achievement versus 50% for trough. And here we're thinking of troughs of 15, okay? So the most important thing that we can learn from here is, well, if we're gonna chase troughs of 15 or higher, then 1 out of every 3 child, is it 1 out of 40? Yeah, 1 out of every 3 child, we're gonna be increasing the dose because we're chasing the trough unnecessarily. Because they have already achieved the AUC. But since if we're going to chase trough, we're going to increase the dose, hence putting the patient at more risk for toxicity, and which we have seen a dose-related toxicity with vancomycin. Just pass on this. This was the model that actually we derived that explains clearance and volume. We did an internal validation with this, but I do want to note that there was a separate group, independent group, that actually used this model to dose their patients, and they were able to externally validate our model from this. So based on all the studies that we've seen in the past 5 years, is a slide I put together that kind of put the range for what the empiric regimen would be for vancomycin use. So for pediatrics, and depending on serum creatinine baseline, a little bit age and MIC distribution, we're talking about a range of 60 to 80 milligram per kilo per day. And traditionally, vancomycin's been dosed more closer to 40 to 50. So we definitely see an increase in the need for the higher doses. For renal insufficiency, because vancomycin is cleared renally, dose is a little bit lower and potentially dosed every 8 hours rather than every 6. There's some recommendations for obese patients, but this really needs further evaluations. Higher doses may be needed for patients with solid tumors. And of course, we need more studies to validate that. And these are the recommendations for neonates and infants based on postmenstrual age and serum creatinine or birth weight, depending on which model we use. So what about effectiveness? We know the AUC and MIC of 400 is an efficacy surrogate for adults. Is that the same for pediatrics? Well, there has been some publications that looked into the effectiveness, not the efficacy. But a lot of these studies with the findings, major findings listed there, were not as strong in terms of study design. All of them were retrospective, first of all, observational, it was non-interventional, it was not controlled, and sample size were very limited. So there's still a lot of work to be done in this area to making sure that we can link the PD surrogate, efficacy surrogate to AUC to MIC. From the toxicity side, we've done a better job in pediatrics. We do have at least 4 or 5 different groups who've looked into this. And you can see here, the incidence varies quite a bit depending on the institution. And it ranges from 4 to 40%. So that's quite still a big gap. But one thing we can learn from all of these studies is that with increased trough concentration, so this is what the guideline is recommending, we're definitely seeing an increase in nephrotoxicity. So there is a dose exposure relationship. In a study that we published, which did use POP-PK with Bayesian estimation, we were able to illustrate that here. So when the initial trough concentration, meaning the trough that you get within the first 72 hours of therapy, the higher you get, the increase in the occurrence of nephrotoxicity. In fact, using multivariate analysis, when we adjust it for stay in the intensive care unit and the use of concurrent nephrotoxic drugs, we were still able to see a 3-fold increase in the incidence of nephrotoxicity. And based on trough concentration of 15 or more, and also we were able to link this to exposure. So when you're targeting AUC of 800 more, which you would if your MIC was about 2, then you're putting the patient at risk for nephrotoxicity. And so this is why we recommend vancomycin you can use, but for MIC of 1 or less. And here are the recommendations from the 2009 National Guideline, again, focusing on targeting troughs. And again, this was mostly from adults with limited information in pediatrics. With the new guideline, which is in progress and expected to be out sometime this year, Dr. John Bradley and I were involved in helping at least write the pediatric section to this. So this guideline will be endorsed by the Pediatric Infectious Diseases Society as well, and we'll be integrating all the literature that we've found in neonates and pediatrics. So the new recommendation is actually going towards an AUC-MIC based on a certain method, the broth microdilution method of estimating MIC of 500 to 650. But still, we don't have any outcome data at all in pediatrics with this recommendation. This corresponds to, if we were to use the e-test, about 400. Okay? It's just that most institutions don't necessarily use e-tests, so they wanted to have some recommendation for the one that we use. I think we use microscan here for the broth microdilution, but this would be the same for the Vitek, et cetera. And the recommendation will be, let's move away from trough. We're actually going to be looking closer at AUC monitoring. And this is going to be consistent for pediatrics, as I have demonstrated with the literature that I presented, as well as adults. So we're moving towards just plain AUC monitoring. So if that was the case, how do we monitor AUC then? One of the other highlight of the guideline is actually to monitor AUC through Bayesian estimation. And we all know what Bayesian means. What this means clinically when we're doing this is to actually we can't unfortunately do Bayesian estimation in our mind. I wish we could, but we can't. So we're going to have some computer help. And I know at Rady Children's we do have TDMS, although there's a new program name for that now, that does integrate Bayesian estimation in the software. So the guideline will emphasize that. And for adults, they're actually recommending using a single trough to estimate the AUC, but using Bayesian estimation for that estimation of the AUC. Now for the pediatric, if we only have a single trough, fine, but we're actually recommending a peak and a trough if it's available based on a study that we published that I'll share with you in the next slide. Oh, one thing with Bayesian estimation is known to be superior when it comes to estimating exposure compared to non-Bayesian estimation, because non-Bayesian estimation tends to underpredict AUC. So you can imagine if you're underpredicting AUC, you're going to increase the dose unnecessarily again. And it's about, I think, 20 or 30% underestimation. Okay, so here's a study that we published, again, using patients at Rady Children's. We were able to demonstrate with PopPK and Bayesian estimation, and we had over 138 subjects for this, that if we were to get a peak and a trough versus a trough alone, we actually improved the accuracy and the precision of estimating as well as predicting future AUCs. And this was the same thing that we saw for estimation of clearance as well. So there is some data out there that peaks and troughs with Bayesian tools would be preferable. Okay, the last segment that I wanted to do was to bring in a case where we can kind of start bringing in what we've learned from research and bridge that to how does this affect our clinical practice going forward. So we have a case here, a 6-year-old boy, 16 kilo, who's previously healthy, admitted to the ICU for right upper lobe pneumonia, which was confirmed by chest x-ray, and the patient did require mechanical ventilation. Labs are listed there with elevated white count of 22, 80% neutrophils, 11% bands, serum creatinine at a 0.40. So the patient was started on ceftriaxone and vancomycin. Vancomycin dose at 210 IV every 6 hours. So what do you think about the vancomycin dose? So given the current dose, the patient's getting about 52 milligram per kilo per day. And so we look at the dosing, that empiric dosing slide that I have. For this patient, the recommended range has been between 60 to 80 milligram per kilogram per day, which is pretty close. So why was the patient started on 52 milligram per kilo per day? Well, that's because we have pharmacy involved at the beginning to precisely recommend a dose. And this is, to me, the ultimate precision medicine example that you can get here. So pharmacy was involved. So we went through a therapeutic drug monitoring using the model that we published here on volume and clearance. We were able to estimate a dose using the empiric regimen. And with that dose, we are targeting actually an AUC of 400. So we are achieving that. So this is very appropriate. Okay, so we always want to use the minimum effective dose. So kudos to the pharmacy who did, who did this. And so 2 days later, the valve culture came back of Staph aureus, specifically MRSA, as you can see here. And the vancomycin MIC through e-test was a 0.5. And After the fourth dose, which represents we're getting at steady state, or in steady state, when we drew these concentrations, we got 2 vancomycin concentrations of trough of about 0.6 and somewhat a peak, which was drawn at 1.4 hours after the beginning of infusion, and that was 6.5. So what do we do here? We got pharmacy involved again, saying, here, figure this out. And so this is what happened. Knowing that the AUC is equal to dose within that 24-hour period, not just the dose for that particular single dose within the day, is dose divided by clearance. So using the Bayesian estimation here, you can see that the current dose that the patient was on, 52 milligram per kilo a day, gave an AUC to MIC, because we do have an MIC now, of 279, which was kind of below what we want. So this is what you would get, the AUC that you would get for Bayesian estimation. Now if we were to use non-Bayesian estimation, how would this look? Using this, remember non-Bayesian only relies on the 2 concentrations that we have and just get an estimate from there. We can see that this underpredicts AUC quite a bit with the non-Bayesian side. And so what we did was we revised the dose to about 310 milligram IV every 6 hours, which comes out to be close to 60 milligram per kilo per day. And with this, we have about an AUC and MIC of 411 using Bayesian, versus we'll be even going higher if we did not have the Bayesian tool. Now, thanks to Bill, who told me to add this slide in. So how would it look when we change the dose, either change the dose or change the frequency of administration? How would the AUC and trough concentrations look? So here, the patient started from 13 milligram every 6 hours, right? And then we increased it to 19 milligram per kilo every 6 hours. So we only increased the dose without changing the dosing interval. So if you go back, looking at the equations of AUC versus trough concentration, you know AUC is only dependent on the total daily dose. And so because we increase the dose, of course we're going to change the AUC. We're going to increase the AUC. Now for trough concentration here, because we increase the dose, we're going to see an increase in the trough with the change in the regimen as well. Okay? What if we kept it at 52 milligram per kilo per day, but changed it to every 8 hours? So here we're changing not the dose, but just the frequency. How would the AUC and the trough change? Would the AUC change? Not at all, because the total dose is the same per day. And so the answer is in brown or yellowish here. So that wouldn't change at all. Would the trough change? Absolutely. And this is very, very important to say, because sometimes, people may assume that when trough changes, the AUC will change too, not necessarily if the total daily dose is the same. And it just goes back to these formulas. So having those memorized should be good. Or have your pharmacist memorize it. Okay, so in conclusion, the research and practice of clinical pharmacology is very critical to optimize dose in pediatrics, especially with the idea of what precision medicine should be. And again, we learned 3 main tools of clinical pharmacology today: POP-PK, Bayesian estimation, and Monte Carlo simulations, which are recognized by the FDA for evaluating drug in pediatrics. And specifically for vancomycin, start monitoring AUC, think about Bayesian tools for the estimation, and really what you want to ultimately get to is the minimum effective dose. So if your AUC is higher than 800, think of alternative agents. And if your trough is greater than 15, move away from that, because you're putting the patient at risk for nephrotoxicity. What do we need in the future? Definitely a lot more PK/PD guided research in pediatrics for a lot of drugs that we use, because we're still learning. And specifically for vancomycin, we still need the PD efficacy, effectiveness studies to make sure that AUC to MIC 400 is really what we want in pediatrics. And this could vary with neonates and the older kids as well. And so far, the guideline for these of vancomycin has really affected the MRSA, but we're seeing this apply for the stag quag negative staph, which is an important pathogen in neonates specifically. So does that apply to that staph as well? And the recommendation, the PD exposure may vary with the different types of infections as well, endocarditis versus meningitis, etc. I wanted to conclude with just special acknowledgments of my collaborators who the research would not be even possible without their support over the years. Starting with John, I'm going to see him later, and I'm going to ask him why he wasn't here. And special thanks to Gail and Bill Pharmacy to really help gave me all the PK stuff that they have over the years collected. And Michelle, who was from microbiology, who was able to provide the isolates that we needed to do further e-testing for the study here. And of course, my colleagues at Miller Children's Hospital, and of course, Edmund and Victor, always from UCSD. Okay, any questions? Yes.
So I was curious when the clinical example that you showed, you said you had pharmacy involved and you calculated the AUC and things like that. So you're basing that on your formula that is already developed, right?
Correct. Well, yes, for empiric therapy, it was based on the formula that we developed. But in terms of the dose revision, as long as you have a Bayesian calculator or tool, that should do well as well. And we do have that here.
I don't understand then the difference between the two formulas, that how could you adjust, what did you do differently in the two formulas to make that correction?
There are different tools that actually use different formulas at their baseline. I know with TDMS, it's kind of fared very well for the patients here, and it does integrate that Bayesian tool. And I'm not sure what specific model is used. Do you know what is used, Bill, for TDMS, for the POP PK side, the empiric dosing? So,
we don't use that formula there.
So the nice thing with the TDMS tools here, with Bill being involved, he can actually do his own mini study on the patients that you have here. So it's definitely much more applicable to your patients here. So that was more generalized to other hospitals as well. We had 2 pediatric hospitals. So that can be used, but it really, this is why you have to assess the program that which has which model. And there's a neonatal model, there's a pediatric model, and in certain softwares that being developed recently, this model was used for that software.
It depends a lot on the inputs you put into it in terms of developing that model, right? In terms of how many overweight kids, how many kids with renal
Yes,
deficiency.
absolutely.
And so say you had an extremely obese kid who you wanted to give vancomycin to.
I would not use this model.
Right.
Because while this is the most generalizable model that you see published, because we had over 700, I think 702 patients and thousands of samples from this. So this will be the most diverse study that you see out there. And I think in our patient, we had about 20% overweight, obese kids, but I would not necessarily use this.
You would go back to your traditional, get the peak and trough and try to.
Well, you still need to start out somewhere.
Right.
So you still, and this, we do have a model that we're trying to create for obese subjects. This is the same thing with patients on oncology. They may need higher, and there are several studies that show that. But this would be the start. And then once you get that, then you get this patient-specific data with their exposure, how that's like through the concentrations, and you integrate both of them.
Are you helping Sandy Benjamin with their bank? Are you doing a Banco model for obese children? Do you know?
We published a paper
Yeah.
that talked about Venko model, but we just didn't take a step further to say what the dose would be yet. But that's in the works at this point.
Yeah, because one of the big networks in pharmacology is the Trials Network. And so they're trying to develop these models specifically, like they're asking us all these group of these children with particular drugs to do exactly what you're asking. To take it the next step and say, in this special population, how is the model different?
Right. Yeah, how do we adjust the dose?
How do we adjust the dose in this patient, and what weight do we use in all those people questions? Yeah.
Yes, Gail.
I just wanted to point out that thank you for the excellent presentation, but our patient population, our muscle and resistance is I think a little bit lower than the long patient. And we don't tend to, meet or start out on those higher doses here. I just want to make sure that everybody's, clear about that. When needed to the higher doses, but I'd say that probably that one example is a bit of an outlier as far as
Correct.
the levels that were obtained in that patient, and that, again, we tend to really start out more empirically, particular dose, unless they've got therapy, really good at lower. And in the NICU population, we certainly don't start at Q6, it's post-natal, age-driven as well as weight-driven, probably we're going to start. So I just wanted to point out that there was an excellent presentation.
Thank you, Gail, for pointing. This is why this slide is very important, because the first thing I mentioned here is that it's really institution-based, based on your MIC distribution. And luckily, Rady's been doing a wonderful job. Resistance rate, very good, very low, even based on the E-test, which is the most conservative test. I mean, 15% only of 1.5 or higher is excellent. And so you can achieve your AUC target with lower doses here, but probably not with other institutions with different MIC distributions.
And luckily, we saw that data right away when we were jumping on the bandwagon for achieving the higher troughs, and we were seeing acute kidney injuries and right away, and so then we all stuck the academic credit bill for meeting with ICUs to make sure that we were using a sensible approach and getting our goals, getting our desired outcomes without giving AKIs.
And one thing to note too is in our patient, when we started the patient on 50 milligrams, that's below what we found in the study, but it was fine for that patient. So really bring it back to the patient and listen to your pharmacist.
Thank you.
Okay, thank you.